to now invite the guest speaker. The guest speaker will make presentation on challenges and prospects of DNA investigation in litigation. To do justice to this paper is no other person than Dr. Richard Somiari, the Director of Lagos State DNA and Forensic Center. Doctor, to the microphone, sir. A round of applause to him.
When I was invited to attend this program, I didn't know it existed, and I apologize. Which is why the awareness is very important. So I asked my assistant to do some background check, and she came up with a lot of information about the association, which was very impressive. So I'm honored to be invited. What I'll try to do is demonstrate that forensic science is needed. There are a lot of people doing a lot of great things that people don't know about. But like we all know, forensic science is a practical issue. It's not about talk. You have to reduce it to practice. And some of the problems we have in Nigeria is not that we don't know what to do, or we don't have the opportunity to do them, because we do not have the tools and we do not have the funding, which can be a problem. So when I got the privilege of being invited to help set up the Lagos State DNA and Forensic Center, one of the things that I struggled with was, why hasn't certain things been done? We attend conferences, we attend workshops. Why are we not reducing them to practice? Big challenge. I can assure you that at the end of this program, you're going to have a lot of ideas. You're going to be so excited that you want to start doing things. But how are you going to be able to do that? It's going to depend on a number of issues, most of which have been discussed by the presenters, the earlier presenters. So, are we ready? So I'll try to challenges and prospects. I don't need to remind anybody of all the challenges we have in Nigeria in all sectors. But like the Prof mentioned, forensic can help. But for it to help, we have to apply it. Okay, good. So I'll start by acknowledging, standing on the existing protocol and acknowledging the organizers and the participants. Okay, they're now in business. So I just thought, because it's a workshop, I know that there are a lot of professionals here, and I might be making it a little too simple for some people, but I'll try and make it as interesting as possible for everyone. So I thought I should define some keywords that I got out of the title of the presentation. So DNA, we are all familiar with DNA, especially in the context of paternity and biological relationships. So it is a self-replicating material that is present in nearly all living organisms. Investigation. There's nobody here that does not understand what investigation is and how powerful the outcome of an investigation can be during litigation. So it's a formal and systematic action taken to examine something, someone, or an incident. And then litigation, process of taking legal action to resolve a dispute, convict, seek redress, or compensation. Challenges in the context of this workshop is to think about the difficulty in undertaking investigations in which forensic evidence could help with litigation. It's very difficult in Nigeria for a number of reasons. And prospects. It's not all bad. What's the probability or chance for future success? I think that's what this conference is all about. I might as well just step down because I've finished my presentation. Any questions? Okay, so let's look at several scenarios. Remember, this is a workshop, right? So for some people, this will be elementary, but there are many other people who are trying to reach. So, this is a scenario. This is a real scenario. How many people recognize this photo? Ikoyi building collapse. That's what it is. Okay? So scenario 2. 2 vehicular accidents, a handgun recovered from a trash can, a knife with reddish stain found in a car, a hotel room with brownish stain on the floor. These are actual images from cases we are working on in Lagos. Okay? A golf ball with a reddish stain found at a golf club. A lady sitting outside a home looking very distressed. So the question is, which is not a crime scene? And in which will forensic analysis be helpful? Any takers? Well, they are all crime scenes. They are all crime scenes. So how do we deal with such scenes in Nigeria? It is only the work of associations like this that will increase the awareness and encourage people to do the right thing. But if you don't know what to do, you're not going to do anything. Sometimes you think you're helping, but you're creating problems. So as I walk through this presentation, I'm hoping that I'll be able to touch on each of these and how it will help. So anything that yields evidence is a crime scene. Could be a location, vehicle, object, body, or tool. A single incident can result in multiple crime scenes. If a person commits a crime and leaves the incident, they say he becomes a walking crime scene. What do you need? Forensic science is expensive. It's an expensive business. Some of the disciplines require very expensive equipment, and the Lagos State government spent a lot of money to set up the lab in Lagos. You need everything because you don't know what you will encounter. No two crime scenes are exactly the same. Experience matters, but this crime scene will be treated as a different, unique crime scene. So the sole purpose of forensic is to use scientific methods to conduct investigations to help in arresting, convicting the guilty, and exonerating the innocent. So there's a process. But I always ask the question, when you have all these tools, all the experience in the world, what can you do to resolve a case? Well, it is what the law allows you to do, right? And you cannot commit a crime in an attempt to solve a crime, which creates significant problems if people do not understand what they can do and what they cannot do. Everybody in this room understands that you want to try to establish that evidence triangle, which is the linkage between the suspect, the victim, and the crime scene. The primary purpose of forensic science is to identify and compare. That's it. And depending on the discipline, you might need very expensive equipment, which is why talk is easy, but reducing it to practice is the key. And unlike other disciplines, forensic science requires a lot of money. I don't pretend about that. So when judges question the results, it's because they do not understand the power and because they do not believe that they are using the right tools to do the right testing. So we have to figure out how to convince them. Now, if you're lucky, you can link multiple crime scenes with forensic evidence. And then, this, when successful, the evidence may find its way to the courtroom. The purpose of forensic scientists is not to find somebody guilty. We all know we must be impartial. We're going to do the test, we're going to give you the result. What they do with the result in court depends on the jury or the judge. That's what we stand for. It's science. Science doesn't lie. So, it's an extremely powerful process. You can use forensic science to establish the elements of a crime. When you stop somebody and you search the vehicle, if you have a warrant, and you find a white powder, what do? You need to be able to establish that it is a banned substance. That's the only time you should be able to arrest the individual. So you can use it to establish the evidence of a crime. You can include and exclude suspects. There is a time limit to which you can hold somebody without formally charging the person. And forensic science can help you resolve that fast. You can use forensic evidence to reconstruct the crime. When you look at the justice system, forensic has power all through the critical steps, from the arrest and clearance stage right down to the sentencing phase. At the arrest and clearance stage, forensic science could help you increase the number of people that are formally arrested or that are cleared. At the charging stage, you need scientific evidence before you can formally charge a suspect. With forensic evidence, this could lead to a reduction in the number of plea bargains. What you don't want to do is to settle by plea and then let the person back into the community to go and continue causing the same problems. Forensic evidence is one of the most trusted type of evidence. When you have physical evidence with science backing it, it's more trustworthy than eyewitness accounts. It is also more persuasive because you can show photos, you can demonstrate. That leaves a lot in the mind of the jury and in the mind of the judge. It will help yield convictions because it will most likely interact with other types of evidence. Linked to longer sentences. When you show graphically the murder scene, that leads to longer sentences. And I have an example here. Well, this is not the biology class, but I thought I should introduce to everyone what this wonderful acronym stands for. Deoxyribonucleic acid is a self-replicating material found in almost all living things. It's mainly located in your nucleus. It's organized in chromosomes. The chromosomes are made of the DNA threads. It's a hereditary material in humans, and it determines specific structure and function of every cell. It's responsible for passing character across generations and traits. Half comes from your mom and half from your dad. 99.5% will be similar amongst us. It's only 0.5% that is different. And it is this difference that we target when we try to use DNA in different ways. There's a unique way that DNA codes, and this is based on some chemical bases known as adenine, guanine, cytosine, and thymine. There's a unique way they pair. A will always pair with T, and C will always pair with G. This is very important. So if you know one strand of DNA, you can deduce the second strand. This is important. And it is not the sequence of DNA that matters. It is the order in which they are organized. So if you think about it, in the English dictionary, we have just 26 letters, right? But we have over 180,000 words. So if you consider TOSP, the reason why this step is important is because people wonder, how do we perform the magic to pick an item of evidence somewhere else, extract DNA, and link it to a suspect? It's because of the sequence that we find, which is highly conserved. So with TOSP, you can form stop, spot. What else can you see? There are people that are good in Scrabble. Post, opt. So just with 4 letters, we have over 3.1 billion bases organized in different sequences. Same thing like the English dictionary. So that's the trick about DNA. So how do we collect DNA? Well, it depends on the purpose. For reference samples from the suspect, you collect the blood or the buccal swab. But on the right side is where the action is, which is where forensic science and experience really comes in. Because we can also extract DNA from containers, from cigarette butts, you name it, from multiple sources. It is this evidentiary DNA that will be compared to that of the suspect. We do not try to sequence the entire human genome, no. We look for specific regions that are repetitive. These are called short tandem repeats. So it is these regions that will be amplified. And if you get, for example, in this example, the mom contributed 7 repeats. So each column is a repeat, whereas the dad contributed 8 repeats. So at that particular location in the genome, this is going to be very unique and it will be passed across all generations. That's how we can establish relationships. All forensic disciplines are important, but it's only DNA that is so sensitive and gives you the statistical likelihood that this is the individual. DNA is also one of the only forensic tools that can be used as a surrogate to identify somebody that committed a crime. What do I mean? My brother commits a crime, okay, but you can use my DNA to identify him. You cannot use any other forensic tool to do that. So, this is where the amplification and sequencing takes place. Thank God for COVID. Everybody now knows what PCR is. So what is PCR? This is polymerase chain reaction. It's a method of amplifying a signal, turning 2 DNA molecules into 4. By the second cycle, it becomes 8 in that order. So you touch a pen, you don't leave any finger mark, we can extract DNA. We call that trace DNA. So it complements biometrics by doing what no other technology can do. Why? Because we can amplify. Yes, you can try to enhance fingerprints, okay, but not at the level, at the exponential level that you can amplify DNA. So we extract, we quantify, amplify PCR, sequence in an equipment. You then end up with a profile. So what is the role of DNA in investigations? It's a very powerful forensic tool that can help determine who. That's the key. Most crimes are committed by humans directly. So who and where, as well as how. So you can compare DNA from a suspect to evidentiary DNA recovered from a crime scene. Compare the DNA found at different crime scenes to determine if they are from the same source or person. We know that you need a DNA database to succeed. But we have done some great work in Lagos without a DNA database. Why? Because in the first instance, you know that the same person committed the crime. Why? Because the DNA you isolate from the different crime scenes are the same. We can also tell if it's a male or a female. Yes, because we can look at one of the locus and determine that this was committed by a male. Now, in some situations, we can create what we call a DNA dragnet. If a crime occurs here today, after we leave, we know everybody that came here, right? Because we registered for the conference. So we request for everybody to come and give their DNA, and we're going to compare that to the evidentiary DNA found in this room. So that's how we can work around the absence of a DNA database. It's also very useful in disaster victim identification. So we have done a lot of identifications in Lagos, the Otodola Bridge disaster, the Ikoyi building collapse. We did all the identification using DNA. Human remains identification, we doing a lot of projects for the Nigerian military because they keep recovering some skeletal remains that cannot be identified using all other methods. Of course, this is a big one. Many people want to know their biological, if this is their biological children or parents. So, What are the important steps that one needs to take during an investigation to help with the litigation process? You want to protect the crime scene. It's a big problem in Nigeria because whenever there's an incident, everybody, they believe they are coming to help, they are actually making it more difficult for you. But what I'll tell you is, based on the 5 years experience we have in Lagos, in a housing estate, where about 30 vehicles are bungled over a 3-hour period in Kenya. Well, when we arrived, nobody believed we can find anything useful, but we did. When people come into the scene to contaminate the crime scene, there are certain areas you will expect, right, that the contamination is going to be heavier than other areas. So if there was a fight in this room that led to somebody dying, and when you examine the body during autopsy, you notice that maybe 1 or 2 buttons were missing from the clothes. Well, somebody coming to contaminate the scene will not just necessarily look for a button under a chair, right? So the experienced forensic scientists will always pick something up, no matter how bad the scene seems to be contaminated. Because the criminal always makes a mistake. There's going to be a silent witness. So, document. You want to document. So in essence, what I'm saying is, I tell the guys to say, look, the fact that someone else was in the room and several people have visited doesn't mean that we cannot process that scene and lift useful information. And we have example. I have 1 or 2 slides to demonstrate that. So document. You want to document the location. You want to use videos, photographs, sketches, written notes. Collect the evidence. Please. Witness statement is also important because forensic is either going to agree or disagree with the statement. Preserve the evidence. A lot of times, evidence is collected and not properly preserved. If it's not properly preserved, it's going to degenerate. If it degenerates, the result is going to be useful. It's not going to be useful. So you want to protect from contamination. Label all items, maintain chain of custody. This is very important. Number 6 is the big one. That could lead to throwing out all the evidence if chain of custody was not maintained. Collect reference materials. In DNA, we need reference materials. In fingerprint, we also need reference materials. In forensic chemistry, we also need reference materials. So you'll be able to compare the evidentiary item with the source. Of course, submit to the laboratory. Big problem. A lot of people collect the evidence and they don't submit to the laboratory. If you don't submit, it's not going to be useful in court. So, challenges. DNA is relatively stable. However, degradation can occur. And if it degrades, then the result will be questionable in court. What the defense does all the time is to try to question the reliability of the result. And one way to attack the result is to question the stability of the DNA. When was it collected? How was it collected? Where was it stored? How did you analyze it? How did you establish that the data you obtained was accurate? What is the statistics supporting this finding? These are all issues that are raised. Exposure to certain chemicals will cause a breakdown. Handling repeated freeze-thaw cycles will cause a breakdown. So when you're setting up a DNA lab, you want to focus on the humidity. At the lab in Lagos, we have power 24/7. We have multiple redundancies because we cannot afford to lose any item of evidence. Moisture is a problem, it's a big one. So you pick up a piece of shirt soaked with blood, you can't put it in a plastic bag because it's going to be humid and the DNA is going to break down. So what you want to do is to dry it as fast as possible and store it in a dry state or in the fridge. Physical sharing can also cause breakdown. So, talking about the challenges, a number of these have already been described, okay? We do not have sufficient funding. Forensics must be funded. There's no 2 ways about it. There are certain things you can manage. This is not an area you want to manage because you're going to spend a lot of time and effort and the entire data is going to throw it away because something was missing. So let's not pretend about it. It's an expensive field. The question is, how should it be funded? There are different funding ways to fund forensics, but that's not the focus of this meeting. There is still limited experts. Okay? A lot of people have the education and knowledge. You don't learn how to play golf by reading books. You go to the golf course and you hit balls. Forensic is one field where you cannot be an expert, competent and proficient by reading books. Simulation will help, but having access to practical, real-life crime scenes is the key. That's the key. We don't have a system in place that will allow that. I mean, I see young forensic scientists. How many crime scenes have you visited? 0. They have masters in forensic science. So that's a big problem. You need practical experience. You need to have experience going to the court to testify. It's a different ballgame. When you are in the lab, you are the king. But when you find yourself in the court, the prof is laughing. Prof, you understand? That's a different ballgame. So if you haven't gone to court to testify as an expert witness, then you have not. Thank you, Prof. You have not started. But remember, there's always a first time. So don't be intimidated. Those of us that have experience, there was a time we had the first time. And we recognize this. So when we're talking about training, it's not about trying to simulate a crime scene. That's okay when you are an undergraduate, but you need practical experience. There's a big problem. The tools. You need the right tools. There is no substitution. Why? Because the defense is going to tear you apart. When was the last time you calibrated this tool? What is the error rate? Who did the calculation? Show me the data. That's the problem. So I don't pretend about it. When they invited us to set up the lab, I told them it's going to be expensive. Do you have the money? They said yes, so let's go. Lack of crime labs and databases. Forensic scientists collaborate. No single person knows it all. The prof said it also. We need to work together because every crime may be multifaceted. May include animals. You need fingerprint expert. You need psychologists. So we have what I call the core forensic disciplines, which are lab-based, but they rely on other forensic disciplines to be able to resolve the cases. So oftentimes you don't know who you need. People call you up and say, oh, we have this problem, and we want you to come and help us. I say, give me the history first. That will determine the other experts I will invite to work with us to resolve the case. So we do not have sufficient. We need many more. We need many more professionals and many more labs. We also do not have a systematic and coordinated advocacy. Hopefully, this organization will serve as a platform to keep hitting. We need to advocate for certain things and also demonstrate that the reason why we need them. Public awareness. This is what this conference is all about. Limited legal backing. Yes, there are certain things the law will specifically tell you not to do. But then, if we have the right kind of backing and enabling laws, and Lagos State is doing fantastic because we have an annual workshop with the judges and other prosecutors and the defense group. So they're doing a lot of work in that area. At the national level, an organization like this can drive that process. Limited training underscore hand-holding opportunities. Forensic science is still artisanal to some extent. Okay? He has given me the warning. So, we need to have young scientists have opportunities. So we need the academic group. They teach them, they educate them, they give them some experience. But now we need to intern these people with people practicing in the field. They are the ones that will take them and show them, hey, look at that, this is how it is done. So that's what it's all about. We lack that significantly. Lack of commitment. So there has to be commitment. So prospects. They need to exist in Nigeria. That was already mentioned by everybody, and we know it. I can talk about this for the next 2 hours. Success stories exist. Things are happening with the limited resources that we have. How are we amplifying those? So I want to use the next set of slides to just give you an overview of what is happening in Lagos. Most populated city, very difficult, but creates a good opportunity. Within 1 year, we were exposed to every kind of crime you can think of. That's how you get experience. We don't pray for crime to occur. But if it doesn't occur, you don't have a practical experience. It's an ISO lab. Credibility and reliability is important. So the first thing we did was to seek accreditation. We wanted a worldwide accreditation, so whatever data we generate will not be questioned. Storage is critical in forensic science. If you can't store it, don't collect it. So we spent a lot of money and time to get equipment. That's the DNA extraction lab. We will have two workflows. The first is the classical workflow, give you data in about 2-3 hours. This is the rapid workflow, which gives you data in 90 minutes. Some of our staff processing the crime scene. A lot of collaboration is required. You need a lot. If you don't have to like the forensic experts. You don't have to like the police or any of the other law enforcement agencies, but you need them to have access to the crime scene. So we are very friendly with everybody. Just some case statistics. So since we started in October of 2017, we've handled a total of 180 what we call forensic cases and 519 relationship cases, 42% sexual assault. And the next big one is homicide. And burglary is the third. So this here is sexual assault, homicide, burglary, and everything else. When you look at the biological relationship is not only paternity. Okay, so we have court orders, peace of mind legal. People want to go to court because they do not think that this chief is from the lineage that should make him a chief, so they want to remove him, so they go to court. Then police order, and then there's a big one also, child trafficking. We deal with that a lot. They get people at Moritala Mohammed Airport with children, they bring them to the lab, the two children are not related, the mom is not the father, the mother of the children. So we do a lot of relationship work there also. And then one of the things we've set up is the DNA database. You go to crime scenes, you pick DNA, but it doesn't match the suspect. But you know, this is related to who caused the problem. So we put it in one of the ponds. So we have ponds for forensic evidence that we have not associated to anybody, missing persons, unidentified persons. There are people from some of the disasters we have processed. We haven't been able to identify them. Why? Because we do not have a good reference. You might think this is my brother, but the person is not your brother. So it won't match. Now the last case. I'm only able to discuss this because this is a case we took from A to Z. And we don't want somebody to commit a crime, but we also do not want the perpetrator to go scot-free. So this is a very good case. Why? I'll just run through this and go straight to the... So it was a double murder case. In May, maybe you saw the big news, a Danish guy was convicted for the murder of the wife and the daughter. This is that case. I wouldn't have been able to discuss this case if the conviction hasn't been resolved. So we went to the crime scene. What we did, we processed everything, textbook. This is the sketch of the apartment. For those people, for the young ones, this is the sketch of the apartment. So this is the case where the white guy was convicted of murdering the Nigerian wife and the daughter. So when we arrived at the scene, 5 days after the incident. So several people have gone in and out, okay? But they were just going around, they were not processing the crime scene. So the hypothesis, when I got there, my hypothesis was that based on some information of people in this room, room number 9, they claimed that they could hear fighting occurring in this room. They claimed that they opened their door slightly. So the first thing we tried to establish was if there was a clear line of sight between this room and this room, and yes, there was. Okay? The next thing was, this is where the body was found when the police came. But the story was that this is where they found the dead lady and she was moved here. But that was not consistent with some witness account. So we had to prove that the lady did not die in this room. The suspect accepted that he moved the lady from here to here. So my hypothesis was, this is where the lady died. So we had to prove that the body was moved more than once. Okay? We have to prove that the body was moved from this room to this room, and then from this room to this room by the same person. So, after the first visit, we came back in the night because we noticed some selective cleaning. So we came back in the night and turned off the lights and used forensic light sources. So what did we find? We found traces of blood spots here, and then in this corridor, coming in this direction. So the body must have been moved from here to this place. But remember, the body has been cleaned, the floor has been cleaned. We noticed that there are a couple of things, I won't take too much time, but they argued, the defense argued, that after they accepted that we detected blood, they then argued that the individual must have tracked the blood from this kitchen backwards. So we had to prove that no, the tracking was not from the kitchen back to the room, but it was from the room back to the kitchen. And how do you prove that? The concentration of the spot as you are moving away from the source. If there is a wet mark here, paint, you step in it. As you are walking away, what will happen? The concentration will be reducing. Okay, so we said no, it's from there to here, not from here to there. So we squashed that argument. The next one was, well, how did we, I won't take too much time to go into this, but this was a very interesting case. Why? Because we applied the textbook, everything that was described in forensic to resolve this very big case, double homicide. Double homicide. Everything done by Nigerians. Okay, in Nigeria. So those are some of the pictures we took. This is a police guy that was working with us. This is one of the forensic analysts from Lagos. This person right here is me here. This was the second visit before we came back in the night. That's the apartment. Now this is interesting because this white photography is important. Take photograph before you move anything. You see this bottle? The chemical in this bottle was found in the blood of the 3-year-old girl. So that's what was used to suffocate her. Okay? There was no evidence that this was burglary related. The safe was not tampered with. A laptop right here was not touched. So this had nothing to do with burglary. There's the laptop. So this was the line of sight I was talking about. So you can see through. This is the kitchen where the body was found. We demonstrated, the argument, defense argument was that she was, this is a workshop, so you have exceeded. The argument, the suspect, I mean the convict now, claimed that the lady was cooking and fell backwards because of gas. We did not find any gas. Toxicology did not reveal any gas in her system. Okay? And when we took measurements, we realized that if you're standing here, based on her height, she couldn't have fallen down and hit her head here. Okay? And there was no biological evidence anywhere on this fridge that will match her blood. However, okay, so this is the balcony. So there are a lot of things we demonstrated. We demonstrated that this mop was used to clean the floor, and they used bleach. Bleach will mask DNA. Okay? However, what they didn't know is if you suspect that bleach was used to clean, you come back 5 days after, the bleach will have evaporated, the hemoglobin will be there. So just give it some time, lock the crime scene, go away, come back. That's what we did. So this is how we demonstrated that the blood was tracking from the room. This is the master bedroom, back to the kitchen. This is where we also demonstrated blood spots and selective cleaning on this side of the bed. They argued that Well, the cleaner came to clean. Well, you won't clean this part of the bed and not clean the other part of the bed. And we demonstrated blood marks here. We really spent a lot of time here in this room. This is the door. We demonstrated that somebody with blood opened this door and cleaned it up. If you didn't commit the crime, you won't clean it. The absence of evidence in itself is suspect. Okay, somebody was bleeding profusely and there's no blood spot anywhere. It makes you clean. That was the first indication. We demonstrated that he took a shower to wipe himself clean. This was very important in this case. So the house help lives in a boys' quarter that you can only access through the kitchen door. The house help left that night and locked the door from the back. When she came in the morning, she couldn't open the door because somebody forgot a key. So, who was the last person that left this kitchen? The house girl. What did you do? You locked the door and went with the key. In the morning you came back, you couldn't open the door. So somebody on the inside used the key and forgot to remove it. We demonstrated that. This is around the fridge. So her head was here. So if she fell backwards, there was no evidence. This negative 20 freezer that demonstrated that she struck her head there and got the injury that killed her. It didn't make sense. So these are not very good photos, but this is how Luminol will reveal the stain on the floor even after extensive cleaning with bleach. So we revealed all that. But the last set of evidence, how we proved that the body was moved twice. It turns out that the lady beat his hand in an attempt to survive, right? So I asked the police, I said, we need a nightgown. This is a nightgown. So we're going to demonstrate using forensic serology that the body was moved twice. Every nightgown has front and back, right? So if you're lying face down, if somebody gives you, it's going to be on this side. If you're lying face up, they lift you. So with those orientation, we screened her nightgown. And there was a high concentration of his blood around the neck of the nightgown, which will be consistent to what you would expect if someone is trying to choke somebody. Okay? Then, if you are lifting somebody, you will expect to find the blood around this area. That's what we did. So we processed the front and back of the nightgown, and we were able to use this nightgown to demonstrate that the body was moved twice. Now, this is a young 3.5-year-old girl's jumpsuit. So what I'm trying to demonstrate here is that we went to the crime scene 5 days after the incident. We were able to pick evidentiary items that were probative. When we went back to our lab, we spent the next 2 months processing the items of evidence. We collected about 56 items of evidence. And when you consider that a single item of evidence like this would generate this number of sub-samples, then you see why forensic is expensive, especially DNA. A single nightgown, all this number of sub-samples we had to take. So, in conclusion, the court sentenced him to death by hanging. This was all over the news because calls from different people from different embassies to comment. But I don't like to comment on these things. I just want to do my work below the radar. We also have a program. Since 2016, we've been running the Lagos State Forensic Symposium, which is a highly practical program. We encourage people, come and tell us your success stories. Don't tell us policy things, because we have no control over the policy makers. But what have you done? How can we demonstrate it? So it has been a relatively successful meeting. And in conclusion, thank you, sir, for giving me this privilege. I like talking to professionals like this. In conclusion, DNA is a powerful tool in forensic. It's not the only tool that is required in forensic, but it is one tool that cuts across all disciplines. Why? Because it's human beings that are always involved. And when you cannot answer questions using other methods, the question of who, DNA will give you that answer. Challenges include cost, limited times in equipment, contamination issues, adequate storage and preservation of evidence, lack of databases, limited number of evidence analyzed, after collection, preservation of evidence, presentation of evidence in court. You need some expertise to be able to do that. The prospects are good because the need exists. We just have to keep pushing until we get what we want. Start small. We took a small business approach in Lagos. Sometimes we want to do everything at the same time. No, take it one step at a time. Success stories now exist. Thank you so much for your patience.